Marhaban al-Aziz, my dear sisters and my dear brothers. So today is a part 2 of Biology 12th standard unit number 2, first chapter, 5th chapter actually it is. So principle of inheritance and variation, which continue from Mendel's law of inheritance. It was during the mid-19th century the headways was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on garden peas of 7 years from 1856 to 1863 and proposed the laws of inheritance in living organisms. During Mendel's investigation into inheritance pattern, it was for the first time that statistically analysis and mathematically logic were applied to the problems in biology. His experiment had a large sampling size, which gave greater credibility to the data that he collected. Also, the confirmation of his inference from experiments on successive generation of his test plants proved that his result pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposing traits. Example, tall or dwarf plants, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observation and the complexity inheritance in them. Mendel conducted such an artificial pollination, cross-pollination experiment using several true breeding pea lines. A true continuous self-pollination showed the stable state inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one's character, which contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seed, yellow or green seeds, inflated or constricted green, and yellow pods, and tall or dwarf plants. Figure 5.1, table 5.1, which is 7 pairs of contrasting characters traits in pea plant studied by Mendel. You have to look that figure and the characters which are dominant trait and recessive trait. Next, further, here it is characters and contrasting traits. First one is stem height, it is tall or dwarf. Second is flower color, violet or white. Third is flower position, axial and terminal. Fourth is pod shape, inflated or constricted. Fifth is pod color, green or yellow. Sixth is seed shape, round or wrinkled. Seventh is seed color, yellow or green. Inheritance of one gene. Let us take the example of such a hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plants to study the inheritance of one gene. In figure 5.2, just look at that figure. He collected the seeds produced as a result of his cross and grew them in a generate plants of the first hybrid generation. This generation is also called as Filial Progeny or the F1 generation. Mendel observed that all the F1 progeny plants were tall, like one of its parents. None were dwarf, as in figure 5.3. He made similar observation for the other plants of trait. He found that the F1 always resembled either one of the parents, and the trait of the other parents was not seen in them. Mendel then self-pollinated the tall F1 plants and to his surprise found that in the Filial generation 2, some of the offsprings were dwarf. The character that was not seen in the F1 generation was now expressed. The proportions of plants that were dwarf were 1 by 4th of the F2 plants, while 3 by 4th of the F2 plants were tall. The tall and dwarf traits were identical to their parents' type and did not show any blending. That is, all the offsprings were either tall or dwarf. None were in between height. Figure 5.3, just look at it. Similar results were obtained with the other traits that he studied. Only one of the parental traits were expressed in the F1 generation, while at the F2 state, both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either F1 or F2 stage. Based on these observations, Mendel proposed that the something was being stably passed down, unchanged, from parent to offspring through the gametes over successive generation. He called these things as factor. Now we call them as gene. Genes, therefore, are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. Example, they are slightly different forms of the same gene. If you use alphabetically symbols for each, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of this character of height, T is used, capital T is used for the tall trait and small t for the dwarf, and capital and small t are alleles of each other. Hence, in plants, the pair of alleles for height would be capital T, comma, capital T, small t, or 2 small t. Mendel also proposed that in a true breeding, tall or dwarf pea variety, the allelic pair of genes for height are identical or homozygous. That is, capital T and small t, respectively. Capital T and small t are called as genotypes of the plant, while the descriptive terms tall, dwarf are the phenotypes. When would be the phenotype of a plant had a genotype capital T, small t? As Mendel found the phenotype of the F1 heterozygote capital T, small t to be exactly like the capital T parents in appearance, he proposed that in a pair of dissimilar factors, one dominates the other, and hence it is called as dominant factor while the other factor is recessive. In this case, capital T is dominant over small t, that is recessive. He observed identical behavior for all the other characters, traits, pairs that he studied. It is convenient to use the capital and lowercase of alphabetical symbols to remember these concepts of dominance and recessives. Do not use capital T for tall and small d for dwarf, because you will find it difficult to remember whether capital T and capital D are alleles of the same gene's character or not. Alleles can be similar, as in the case of homozygous capital T and small t, or can be dissimilar, as in the case of the heterozygote capital T or small t. Since the capital T, small t plant is heterozygous for genes controlling one character, it is monohybrid, and the cross between capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to a gamete. This segregation of alleles is random process, and so there is a 50% chance of gamete containing either allele has been verified for the results of the crossing. In this way, the gametes of the tall plants have the allele capital T and the gametes of the dwarf plants have the allele of small t. During fertilization, the two alleles capital T from one parent, say through the pollen, and the small t from the other parent, then through the egg, are united to produce zygotes that have one the capital allele and the other is small allele. In other words, the hybrid have capital T small t. Since these hybrids contain allele which express contrasting traits, the plant heterozygous, the production of gametes by the parents, the formation of the zygotes. The F1 and F2 plants can be understood from a diagram called Punnett square as shown in the figure 5.4. You can look at that figure. It was developed by a British genetist, Reginald C. Punnett. It is the graphical representation to calculate the probability of all the possible genotypes of offsprings in a genetic cross. The possible gametes are written on two sides, usually the top row and the left columns. All possible combinations are represented in box below in the squares, which generates a square output form. The Punnett square shows the parental tall capital T male and dwarf small t female plants, the gamete produced by them, and the F1 capital T and small t progeny. The F1 plants of genotype capital T small t are self-pollinated, and the symbol is female and male are used to denote the female X and male pollens of the F1 generation respectively. The F1 plant of the genotype capital T and small t will self-pollinated produce gametes of the genotype capital T and small t in equal proportions. When fertilization takes place, the pollen grains of genotypes capital T have a 50% chance to pollinate X of the genotypes capital T as well as the genotypes of small t. Also, pollen grains of genotype small t have 50% of chance pollinating X of genotype T as well as of genotype small t. As a result of the random fertilization, the resultant zygote can be the genotypes of capital T, small t, or small t, small.